Welcome to the TOEFL Listening section. In this section, you will listen to an academic lecture and then answer questions about what you heard. You may take notes while you listen. You will have time to review your notes before answering the questions. Listen to part of a lecture in a university class. You will hear the lecture only once, so listen carefully. After the lecture, you will answer some questions about what you heard. Some questions ask about the main ideas and supporting details. Other questions ask you to make inferences or connections between ideas. Answer each question based on what is stated or implied in the lecture.
Good morning, everyone. Today we will examine the DNA replication process and focus on the molecular mechanisms that allow cells to copy their genomes accurately and efficiently. We begin at the origin of replication. In bacteria such as Escherichia coli, a single origin called ORIC initiates a replication bubble. In eukaryotes, multiple origins fire to replicate large chromosomes within a reasonable time. At the replication fork, the first major player is helicase. Helicase uses energy from nucleotide hydrolysis to unwind the double helix and create two template strands. Once the strands are separated, single-stranded binding proteins coat the exposed single-stranded DNA to prevent it from reannealing or forming secondary structure. Meanwhile, topoisomerase relieves the torsional stress that would otherwise accumulate ahead of the fork. Replication cannot begin without a primer. A primase synthesizes a short RNA primer that provides a free 3' hydroxyl group for DNA polymerases to extend. This is an essential step in both prokaryotic and eukaryotic systems. DNA polymerases synthesize new DNA in the 5' to 3' direction. Because the two template strands are antiparallel, this leads to asymmetric synthesis. The leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously as short fragments called Okazaki fragments. In bacteria, the primary enzyme complex that performs rapid chain elongation is DNA polymerase III. In eukaryotes, polymerase epsilon and polymerase delta carry out most of the bulk synthesis. All of these polymerases have intrinsic proofreading exonuclease activity that removes misincorporated nucleotides to improve fidelity. After an Okazaki fragment is begun by primase, the RNA primer is later removed and replaced with DNA. In bacteria, DNA polymerase I performs primer removal and replacement. In eukaryotes, RNase H and flap endonuclease participate. Finally, DNA ligase seals the remaining nick to create a continuous phosphodiester backbone. The coordinated ensemble of helicase primase polymerases and associated factors is called the replisome. Coordination is important because the two sister replication forks must move in a synchronized fashion for efficient chromosome duplication. The semi-conservative nature of replication was elegantly demonstrated by the Meselson and Stahl experiment in the 1960s. That experiment showed that each daughter duplex contains one parental strand and one newly synthesized strand. Replication accuracy is not perfect, but it is very high. Proofreading reduces the error rate by about 2 orders of magnitude, and post-replication mismatch repair further reduces errors. This combination keeps the mutation rate low enough for most organisms to survive, while allowing evolutionary change over long timescales. A special challenge arises at the ends of linear chromosomes. Conventional replication cannot fully duplicate chromosome ends, and so cells use a specialized enzyme called telomerase to extend telomeres and prevent progressive shortening. There are important practical applications to understanding replication. Antibiotics, such as quinolones, target bacterial topoisomerases and interfere with replication. Cancer researchers study dysregulation of telomerase and replication stress as targets for therapy. And many laboratory techniques rely on polymerases to copy DNA for sequencing and analysis. To summarize, replication proceeds from origins, helicase unwinds, single-stranded binding proteins stabilize, primase lays down RNA primers, polymerases synthesize new strands with proofreading, primers are removed and ligase seals nicks, and telomerase handles the ends of linear chromosomes. This coordinated process is central to heredity and cell division.
Listen again to part of the lecture, then answer the question.
Conventional replication cannot fully duplicate chromosome ends, and so cells use a specialized enzyme called telomerase to extend telomeres and prevent progressive shortening.
This concludes the listening section. For more TOEFL practice tests and study materials, visit Spinta.org.